Cek, cek. Suara saya akan masuk? So can you hear my voice? Okay, thank you. All right, so let's start now. And hello and welcome everyone to today's webinar session held by Symbiotech in collaboration with BioWiscom Germany. My name is Afif Pranayajati, a PhD student at Monash Biomedicine Discovery Institute, Monash University, Australia, and the CEO of Symbiotech, who will accompany you as the moderator throughout today's session. So today we are gonna listen to an intriguing talk about recent update in synthetic biology and biotechnology, which will be presented by our special guest speaker, Professor Wolfgang Melm. Before we start the session, I would like to inform you today's session rundown. So before we start with the main talk session, I would like to share a short introduction about our company, Symbiotech, and afterwards we will listen to our main talk session by Prof. Wolfgang Melm, and followed by the Q&A session, and then we will close our session with some announcements. So thank you so much, Prof. Melm, for joining us on time. Yeah, nice batik anyway. Okay. Okay, so let me start with the introduction of our company, Symbiotech. I will share my screen. Can you see my screen for participants? All right, okay. So this free webinar is a webinar that's been organized by the company Symbiotech in collaboration with BioWiscom from Germany. So about us, Symbiotech is a first company which working on the synthetic biology, and our main focus is try to give the services for the academia and also professionals in Indonesia who are interested in biology, synthetic biology. And yeah, because now we are entering the new era of natural product discovery. So this is our motivation to establish our company, Symbiotech. So nowadays, since the advance of genomic sequencing technology, we can get a lot of genome data, especially from our country, which has mega biodiversity. So our company will try to help you to explore this mega biodiversity using synthetic biology approach by helping you to give training or maybe workshop with, for example, like genome mining to uncover the potential of genomic data. And from that data, you can produce your beneficial products. And as a country with mega biodiversity, we really know that Indonesia has the enormous genome sequence resources. So that's why we are here to help you to explore this mega biodiversity to the next level using synthetic biology. And there are a lot of potential as well to find novel biological parts. For example, maybe some of participants here has interest in finding novel CRISPR-Cas system, maybe from our country, or maybe other biosynthetic gene clusters that has a potential for natural products. And also we have unique environments that we can explore, like toxic tolerant microbes, which maybe we can find something interesting there, and also extremophiles, or maybe some rare microbes. So this is our main service. So we will provide you a service to gene synthesis and codon optimization, cloning kit, and also molecular device selling. And especially for now, our main focus is on the research consultancy and also training. And also later we will also publish a journal in the field of synthetic biology and biotechnology, which will be launched together in the synthetic biology and biotechnology conference in the end of this year. So this is our services, and you can just visit our website to get more information about our service. And I would like to talk a little bit about our next event, Science Camp program in Berlin, Germany. So this is a program, collaboration program with BioWiscom, especially with Professor Wolfgang Nelm. So this science program, Science Camp program, will be held 1 week for 1 week, and the location will be in the suburb of Berlin, Germany. And the program consists of several trainings, like lab course of CRISPR-Cas, and lab course of DNA fingerprint, lab course of pedigree analysis of PCR, also lab course about DNA structure, and also lab course about photosynthesis. And there are some places that we will visit as well there. For example, this one, Glasser's laboratory that can give a learning session, learning center, and also Campus Berlin-Buch, which is a biotech park in one of the biotech park in Germany. And also we will visit the Max Delbruck Center for Molecular Medicine. And there are also other places that we can visit for the leisure time, like the museum and other landmarks in Germany, like Berlin Zoo, or maybe also the famous Berlin Bomb Museum. And for the further information, just stay tuned on our channel in the WhatsApp group and our website. We will inform you more details later about this program. And if you are interested in it, keep in touch with us. And I would like also thanks to our partner today, Akademisi, and also BioWiscom, especially Generasi Peneliti in Bio Indonesia, and also Komunitas Synthetic Biology Indonesia. So that's the introduction of our company. Okay, so today we are going to listen to an introductory talk about other in synthetic biology, especially CRISPR-Cas modification. So I would like to greet our speaker. Hello and welcome, Prof. Nela. I think you still with mute. Could you please turn on the microphone?
Okay.
Okay.
Microphone is on. Thank you.
Yeah. Okay. Thank you. So, how are you doing?
I'm fine. Just for the introduction, I put on my Indonesian hat.
Oh, really
Is that
nice.
okay?
Yeah, sure. Sure. Very nice.
I take it off later.
Yeah. So it's been a great honor and pleasure for us to have you today in this session. And glad to hear that you are doing well, despite some, yeah, I think there are some terrible news about the weather in Europe recently.
I have to say I like it. It
Oh,
feels
okay.
a little bit like Indonesia.
Okay.
And I'm only at running temperature around 28, 29. And
Okay.
this is what we have around now.
Okay,
So I
interesting.
feel at home. It's fine.
Great. Okay, so before we start our main talk session, let me share briefly the profile of our guest speaker today. So, Professor Wolfgang Millen is a professor for kinetics from Universitat Kassel, Germany. Professor Millen got his doctorate degree from Institute of Genetics at Universitat Düsseldorf, and then spent some years as a postdoc at University of Marburg and University of California, San Diego, or UC San Diego in the United States. Professor Millen also had great experience in science communication and education, as he had been appointed as a director of the Institute of Biology, Kassel University in 2003 to 2006. Was a member of FEBS Education Committee from 2012 to 2015, and also been actively involved in professional societies globally, most notably as president of Genetic Society in Germany, and was a member of American Society of Microbiology and RNA Society, and also been appointed as a lecturer in several universities in Germany, Jordan, Japan, Kazakhstan, and especially Indonesia, as a visiting professor at Universitas Brawijaya, and also spent some time in Universitas Andalas as well, right? Okay, and currently Professor Neil had established a company for science education called BioWiscom, which is our collaborator for today in Germany. And throughout his academic career, Professor Neil had been awarded with some honors and published some influential research articles in the molecular genetics, especially in RNA regulation and CRISPR-Cas study. One of my favorite is the CRISPR-Cas study for public and students in school. Yeah, and recently, Prof. Neil has a great research interest in biodiversity, metagenomics, and also CRISPR-Cas mechanism and diversity. So we are so glad to have you here, Prof. And so now I think it's time for us to listen to Prof. Neil talk about CRISPR-Cas modification, versatile tools in molecular biology. And you can start to share your screen, Prof.
Okay.
Maybe.
I'll try. Let's see. Oops. Oops. Sorry, this starts at the end.
Okay. No worries.
So you can see my screen?
Sweet. Yeah, I can see it. Okay. So
sound is okay?
The sound is okay as well. So just
you
a kind. Yeah. Okay.
Okay.
Okay. So just a kindly reminder for all of the participants. If you have any questions, we will have a Q&A session after the talk. And if you have some questions later, you can also just drop the question in the chat room. And if you feel not so confident with your English, you can feel free. Just feel free to drop question in Indonesia, which later I will try to help you to deliver it to Prof. Neil. Okay, so the presentation is ready. Professor Neil, the time is yours.
Thank you very much, Afif. Thank you very much, Didik, for inviting me. Selamat pagi to all the listeners and participants of this seminar. It's really a great pleasure to be here. And this is not the first seminar I'm giving for Indonesia. It's always been a lot of fun to talk to people, especially in the question and answer session. So let me first briefly introduce. Talk is about CRISPR-Cas modifications as tools in molecular biology. The Bioviscom company, it's a very, very small company. It's based, it has partners. On the right, you see our new partners in biotech. We are also working with the mini-PCR Bio in the US. What Afif already mentioned, Gläsernes Labor in Berlin, actually the place where the planned trip to Berlin is going to happen. Part of what I'm going to present was initially sponsored by or financed by the DFG, the German research funding agency. And it was, well, Bioviscom is an offspring of ScienceBridge, which had worked for about 25 years at Kassel University, and then the university decided to erase it because they thought it was not anymore necessary. At the same time, they also erased all the contacts we had established with Indonesia. That's very, very sad, but well, that's the way it happens. All right. So Bioviscom is a small company dedicated to science communication. We are organizing seminars like this one, workshops, lectures, lab courses, field trips. Except for the field trips, everything can also be done online. Lab courses, well, lab courses is better in the lab than online, but we may also offer some online. And the audience is schools, universities, general public, special interest groups. So especially in Germany, it's very important to include the public to get more acceptance for science and research. The team, the Bioviscom team, consists of scientists in academia and industry. They are all freelance at the moment. We have school teachers, we have artists, we have IT people, and the close connection to Indonesia is expressed by our ambassadors in Indonesia. This is Siti Nur Aisyah from Jogja, Isyatul Aziza from Malang, now in Berlin actually, Baby from Manado, and one of our biology artists, come back to him, Khalid from Jember. So we'll see some of his work later. Oh, this is very important. Indonesia is currently a guest in Kassel. You may not even know about it, but this is, we are currently hosting one of the largest modern art exhibits, the Documenta in Kassel, and this is curated by an art collective from Jakarta. So we have lots of Indonesians right now in Kassel, and everything in the city for 100 days is about Indonesia and Indonesian art and arts from other countries. So it's interesting. We are becoming Indonesian in Kassel. Okay, but now let's go to the real topic. CRISPR-Cas, just to remind you in the first few slides, Emmanuelle Charpentier and Jennifer Doudna invented CRISPR-Cas in 2012. Well, invented is not quite correct. It has always been there, but they found it and they made it applicable. It was actually found. The mechanism was found a long time ago by basic scientists. So it's always a little bit wrong to give all the credit to just two people. There are, I would say, hundreds of people who contributed before they managed, Emmanuel and Jennifer managed to get the breakthrough and make it visible and applicable. So to make it a little bit more simple, what they did in 2012 is they invented the wheel. Now, 10 years later, we are here. So the basics of CRISPR-Cas came out in 2012, were awarded with the Nobel Prize in 2020. And now we are at a high-tech level, and it's still growing almost day by day. And especially Jennifer Doudna contributed immensely to the further development of CRISPR-Cas. All right, so just for those who are not absolutely familiar with the system, let me very briefly wrap up what CRISPR-Cas is. It's a bacterial immune system to protect against phages, and what is mostly forgotten, also plasmids. Plasmids are also, let's say, parasites in bacteria. What is also not so much known is that there are many more functions of the CRISPR-Cas systems, but I will not address these right now. So the CRISPR-Cas locus, let me just see if I can use a pointer, or can you see the mouse? Yeah?
Yeah, Prof.
Yeah, okay. So the CRISPR-Cas locus consists of two parts. We have first the CRISPR array, which consists of little snippets of phages or plasmids that this microorganism has encountered in its previous life, or its parents, grandparents, great-grandparents, whatever. So this is kind of a notebook. This is kind of a library, what enemies, what parasites this organism has met before. Next to it is the Cas locus, which consists of coding genes encoding proteins that are required to process the CRISPR array, to introduce new snippets, and to trigger and to do the job of the immune system. So here is the machinery, here is the library of the enemies. Okay, briefly summarized, what always happens is that bacteria are infected by viruses or phages or by plasmids. To protect themselves, they, using these proteins from the Cas locus, they may cut out little pieces from the virus DNA or the plasmid DNA and incorporate these into their CRISPR array. So essentially, they always incorporate on this site, so you can read the history of infections from right to left. This was a long time ago that this pink virus infected the bacterium, and the green one is the most recent, and a little snippet is incorporated into the CRISPR array. What happens next is that this whole thing is transcribed. So you get a pre-CRRNA, and this is processed into these little snippets again, separated by the black symbol, which is a structural element. So you have many, many small RNAs, which are called CR or CRISPR RNAs, and they associate with one or more of the Cas proteins to make a machine that recognizes, by this little sequence, recognizes a newly infecting phage or virus and cleaves it. Because one of these proteins, one of these Cas proteins, is a nuclease that cleaves DNA or RNA, depending on which protein it is, and thus destroys the infecting virus or plasmid. This is called the interference. So very, very briefly, we have a library of small DNA pieces which are transcribed to RNA, and they serve as guides for a nuclease to eliminate a new infection. Okay, that's very briefly the CRISPR-Cas system. But there is not the CRISPR-Cas system. They are very diverse. And interestingly, many of those have been independently invented several times in evolution by different bacteria. Their function is very similar, their mechanisms are very similar, but it has been shown that they arise independently. So a good mechanism is invented several times. And this is just to impress you. This is the situation in 2019. And here are the different CRISPR-Cas systems. They have all been elucidated and there are more to come. It's an almost endless array of different systems. They have now been subdivided into 6 groups by certain features. We don't have to get into the details of that. It's just to impress you how many different systems, CRISPR-Cas systems, exist. And these are, in a way, relevant for this talk because you can go back to all these different systems which are somewhat different in their action, in their function, though they all do essentially the same thing, to be an immune system. They differ slightly, or more or less slightly, in function, in size, in structure, and whatever, and you can choose among many, many different proteins and systems. Okay. What is of most interest is in the CRISPR-Cas system is the protein with the nuclease function, the protein that cuts DNA or RNA and can be used to modify, to destroy, to interrupt genes, for example. The Cas9 protein is not only the prototype, it's the most famous one, but it's also the nuclease that has been used for most of the modifications. There are now others coming up, or have already come up, but let's concentrate on Cas9 because this is the one which has been mostly used. So again, very briefly, Cas9 is in contrast to some other system, to systems of class 1 CRISPR-Cas. Cas9 is a single protein. a single nuclease. In the class 1 system, you have to assemble a lot of different proteins to get the nuclease function. Here you have one single protein, which is very convenient. What is a little bit less convenient is that in contrast to other systems, Cas9 requires 2 RNAs to become functional. Once the CRISPR RNA, which recognizes the target that is to be cut, and second, a structural RNA, which is called the tracer RNA. So these both RNAs, CRRNA and tracer RNA, hybridize to form a 3-dimensional structure, which is recognized by Cas9. It's inconvenient to have 2 different RNAs. So the first thing that was done already by Emmanuel Charpentier and Jennifer Doudna is to combine these 2 things by a synthetic linker. So now we have only one RNA, the so-called SG or single guide RNA, which makes it easier to work with the system. Okay. What most of you know is how CRISPR-Cas can be used to knock out genes. And this is simply by cleaving the DNA. So you can target essentially any DNA sequence specifically by either synthetically making an SG, single guide RNA, that targets the Cas9 protein to a specific site in the genome. It will then cut there like this, and then you just rely on the endogenous repair system of the cell. Cells encounter cleavage all the time, and you're always in danger. You and any other organism. Your DNA is fragile. Your DNA is being damaged, and therefore cells have developed repair systems. And in some cases, the repair systems are not very good. It's mostly important to rejoin the break point. You don't want half of a chromosome to be lost. So it's better to glue it back together. And in this repair, you get small insertions. It's not exact. You get small insertions or small deletions, which usually disrupt the reading frame of the gene which was targeted. There's one additional point that I would like to mention. In some cases, maybe not even so few cases, the joining may be even correct. That's good for the cell, but not good for our intentions, because we want to make changes. Whenever the joining is correct, the CRISPR-Cas system will cut again, because it still recognizes the correctly rejoined DNA. Only when there are mistakes, the sequence has changed and CRISPR-Cas can't cut again. So you're kind of forcing an incorrect repair. And thus you're forcing a knockout, a frameshift mutation in this gene. Another option is a little bit more complicated. For repair, cells usually look for a template. They don't know exactly, sorry to make cells think like humans, but this is easier to understand. When a DNA breaks, a cell tries to repair as exactly as possible. So it's looking for a template. Where can I find the correct instruction to make this repair? And if you provide something like that, you get homologous recombination. So when you introduce, where's my, here. When you set the cut and you introduce an excess of an RNA, of a DNA that is similar to the site where you cut, you get a crossover, and you may trick the cell and introduce an additional piece of DNA. So you can insert genes, or you can also delete pieces of DNA, pretty much exactly. So that's the basics. This first generation of CRISPR-Cas faced a couple of problems, and people immediately jumped on that to solve these problems. But let me just summarize the problems. Knockouts by deletion or insertion are not really predictable. You don't know exactly if you have if you get an insertion or a deletion, and if you get 3 nucleotides inserted or deleted, or multiples of 3, then you still have a functional protein, but it may be altered in function, and you may not want this. In some cases, depending on the organism, depending on the tissue, whatever, you get off-target effects in that Cas9 cuts at additional similar sequences. This is also not very nice, because you want to control and you want to exactly and only cut at a given site. The second mechanism that I showed you before, the insertion deletion of DNA fragments by homologous recombination, is usually a very rare event, and it is pretty laborious to find these rare events among a lot of unsuccessful samples. And this is, especially in Europe, a very strong point. We have a very strong lobby that rejects any changes in the DNA sequence, even though they cannot be distinguished from naturally occurring mutations. So people just say, we don't want this. When you change a base in DNA, this is by definition dangerous. So we have to look for mechanisms, we have to look for methods, not really because they have to be better, but just to pacify these people. It's kind of ridiculous, but that's the way politics are working. Okay. So what do Cas enzymes look like, and how do they function? That's a very important prerequisite to modify the system. And we asked our artist, Lucas, who is not a biologist, what do you think Cas9 looks like? And he said, well, it's pretty obvious, that's what it looks like. This was pretty close, but not quite exactly. This is what Cas9 looks like. We have by now very, very good structure information on many of the Cas proteins. And I'll give you some more insight here. And I will just point out the things that are important for the following slides in this talk. So this is the structure of Cas9 with the RNA, the single guide RNA here, The DNA, you can just see a glimpse of the yellow here, and here it's hybridized. You can maybe see the yellow and the orange hybridization between the guide RNA and the DNA strand. The domains of the protein are depicted here. HNH is one of the nuclease domains, and RUFC is the other nuclease domain. DNA has two strands, so you have to cut two strands, and these are separate independent domains. I come back to that. The REC domain is a kind of sensor. I don't want to go into the details of that. This one, the PAM interacting domain, I'll come back to that in a minute. So it was crystallography, NMR, cryo-electron microscopy that were used to establish this structure, which is very, very exact and very close to the reality. Modeling, molecular modeling and things like that are, of course, also important to also understand the dynamics. I mean, this is not a stable thing, which is always in this structure. This whole thing moves while it's acting, while it's functioning. And yeah, I'll show you something about that in a minute. Let's just go to this PAM interacting or PI domain, because it's another interesting feature which kind of limits the application of CRISPR-Cas. PAM is the Protospacer adjacent motif. It's a 3-nucleotide sequence, usually a 3-nucleotide sequence in the DNA next to the target site. And this is not recognized by base pairing, but this is recognized by amino acid sequences within the Cas protein. So amino acid sequences, a certain motif in the PAM interacting domain can recognize base pairs, these 3 base pairs. And when that was found out, it was clear you can only use targets that are next to a corresponding PAM sequence. So you can't put your target at any place as you want, but you have to first check for, in the case of Cas9, an NGG sequence. And next to this, you can put your target site and construct a CRRNA or a single guide RNA. But then the people doing the structural analysis came into the picture, and they said, well, we know which amino acids interact with which nucleotides. They changed the PAM sequence, and now we have different Cas proteins that recognize different PAM sequences. So that's very convenient because you can now target almost any sequence in a given genome. Yep. So changes in the PI domain result in recognition of different PAM. And so Cas9 has been modified to recognize different PAM sequence and extend the range of targets. As I said before, we can look at the dynamics. Mostly this is done by modeling of structures. I'll try to do this and show you that I hope this works. Let's see. Does it work? Yes, it does. So what we have here is a scanning force microscopy movie of Cas9. Can you see it?
Yeah, sorry, Professor, we still cannot see the video.
You can see the video?
I cannot see it.
Oops.
So maybe you have to switch the screen a little bit to the Facebook. Person.
Whoops. I don't really know. Nobody can see the screen? I can see it.
Yeah.
Oops.
Okay. I think you can just switch your screen. You can stop your screen probably for a second and then start share screen again with the different.
Okay, I'll try that.
Yeah.
Okay. Let's see if it works.
Yeah. Yeah, now it works. Yeah. Probably,
now it works.
Yeah, we can see the Facebook.
Okay.
Yeah. Okay.
So
nice.
What you can see here is the Cas9 bound to a DNA. You can't see the single guide RNA, but what you can see is the cut. Let's look at it again. So it wobbles, it moves, it changes structure all the time. And then at some point it cleaves the, let's see, oops. I have to go back to the,
yeah.
Okay. Sorry, this is a little bit complicated.
No worries.
Oops. Okay, oops. Yep. So, okay, so what you can see, this is actually a live movie of CRISPR-Cas in action. And I thought that was, when I first saw it, I thought it was extremely impressive. Okay, now understanding the structure. What we have now is the first modifications. As I said, oops, I have to go to this. Yeah. As I said, we have two nuclease sites in the Cas9 protein. The RUFC cleaves here, the HNH cleaves here. So you get a double strand break. This is what Cas9 usually does. Now, engineering the protein and understanding these cleavage sites exactly, what you can do is eliminate one of these sites, like this, just change the amino acid, and what you get is a nickase, N-Cas9, an enzyme that, a Cas enzyme that just cleaves one strand and not the other. So you don't get a double strand break, you just get a nick in one strand. We'll see later why this is good. If you eliminate both sites, you have a dead enzyme, which is called D-Cas9 for dead Cas9. What do you want to do with the dead enzyme? To put it in the garbage? No, it's useful. It still targets a specific sequence, but it doesn't cleave there. We'll see in a minute what you can use it for. So keep in mind, we have N-Cas9, the nickase, and we have dead Cas9, the non-cutting enzyme. What you can use the dead Cas9 for is, for example, to visualize sequences. When you have the functional Cas9, It will cleave the target site and then disappear, will diffuse away. While dead Cas9 can't do its job, it will just sit there and stay. Now, you can do furt' engineering and attach a fluorescent group like ATO 550 to the 3' end of your single guide RNA. Or you can attach a fluorescent protein like GFP to the Cas9 protein. And using this in a cell, you don't get cleavage, but you get fluorescent at specific sites. So you can use it to visualize specific DNA sequences in situ. And what you can see here is the pink label is centromeres of a plant chromosome from Andreas Huben at the IPK in Gattersleben. So far, this can only be used for repetitive sequences like centromeres, telomeres, and other repetitive things because the signal is still too low for single copy genes, but people are working on this. So this is one point to visualize sequences. The next one is blocking gene expression. And again, we are using dead Cas9. And I don't have to remind you, just for the fun of it, we have a coding gene, we have promoters, and promoters bind transcription factors to get gene expression. When you now target a dead Cas9 to the promoter, you get this. Promoter is occupied by the dead Cas9, sequence specifically, just this promoter and no other promoter, and the transcription factors can't bind. So essentially, you're downregulating or knocking out the gene without interfering with the sequence. So transcription is partially inhibited essentially by a roadblock because the factors cannot bind. What is interesting in this context and also in others, the binding, and I'm not going into the details of that, the function of Cas9, of the dead Cas9, can be made inducible. So you have the Cas9 in the cell, and it will only bind when you induce by chemicals, by light, or by whatever. So you can make it inducible, and only the roadblock will only work then. This blocking of gene expression can be improved. You can make it more safe, more reliable by attaching something to the dead Cas9. And transcription inhibitor, for example, like the CRAP protein. And Cas9 is such a small protein, and you can attach all kinds of things without interfering with its function. And when you attach a transcription inhibitor, you not only have a roadblock, which may be eliminated by very strong binding transcription factors. Here we have a functional interaction with an inhibitor on the promoter and stop of transcription with of the coding gene. This is called CRISPR-I, CRISPR inhibitory. But you can also do the opposite. You can attach a transcription activator like GCN4 or BP64. Such activating domains attached to to Cas9, again the dead Cas9, of course, and you get an increase in transcription. You enhance gene expression. This is called CRISPR-A, CRISPR activation. And again, these regulators can be made inducible. Let's go to the next modification. Epigenetic changes. Genes can also be regulated by epigenetic DNA modifications. The best known is 5-methylcytosine in symmetric CPG sites. This is CPG, C phosphate G, C phosphate G, and an enzyme attaches methyl groups to these cytosines on both strands. This is why it's CPG, because on the other side you have CPG in the same orientation. Okay. This methylation mostly sits in promoter regions and usually results in turning down promoters. When they are methylated, they are turned down. Again, what you can do, you can attach a methyltransferase like DNMT3 to the Cas9, again, the dead Cas9. You don't want to cut the DNA, you just want to modify it. Methyltransferase attached to Cas9 on a flexible linker. And what happens is you target this to a promoter region or to any other region that you want, and the enzyme methylates the DNA. So again, you interfere with gene expression without changing any sequences. You're just changing modifications, and this is reversible. So these, depending on the application, these epigenetic changes could be transient, permanent, or again, inducible. Diagnostic tools. And here we come to one of the other nucleases in CRISPR-Cas systems, and this is Cas13a. As you may know, or you should know, is that you do not only have DNA viruses, you also have RNA viruses. And Cas13 is something, is an enzyme that cleaves single-stranded RNA. It also requires a CRISPR RNA to find the target. And Cas13a has another feature, which is, well, a bit strange, because if there's no target, this is normal. If there's no target, the protein is just sitting there and not doing anything. When a target is recognized and cleaved, then you get promiscuous cleavage of all other RNAs. So essentially, the cell is killed by cleaving all the RNAs. This is okay, this is fine, because better to kill a cell, to kill a brother or sister, before this one cell produces a lot of viruses, phages, whatever, and destroys the entire population. Now, this Cas13 system can be used as a diagnostic tool. When you have a patient sample, you mix in an indicator RNA. Indicator RNA has a fluorophore, which is attached synthetically, and on the other hand, a quencher, which blocks the fluorescence of the fluorophore. Okay, now you take the patient sample. Let's say we have a patient that has no viral RNA in, and nothing happens. Now you have another patient that has viral RNA. This is recognized. You get the first cleavage. And after the first cleavage, Cas13a becomes promiscuous and cuts everything, including the mixed-in indicator. And here you separate the quencher from the fluorophore, and you get fluorescence. So this is a very, very sensitive system. When you have a viral target for the Cas13, you get promiscuous cleavage and you get fluorescence by your mixed-in RNA. This system has been developed. It's called Sherlock, and it has been developed as a diagnostic tool for SARS-CoV-2, but also for other viral infections, because it just depends what CRISPR RNA you're including into the Cas13 complex. Another application, which is, I believe, fantastic, it can be used on, or it could be used on humans. It's not, I don't know if the medical trials are already on the way. It's a strategy which was adapted from the mRNA vaccines. And here we are talking about LNP, lipid nanoparticles. And they include the mRNA of Cas13a and a cRNA as an address label. When you introduce these into a cell of an animal or a human, the mRNA is transiently translated into the Cas13 protein, and the address label, the cRNA, is incorporated into the Cas13a and is now looking for a target. These lipid nanoparticles can be vaporized to form an aerosol. And what you can do is, and I show this because, or this is done by Lucas, because the experiments were done on hamsters. Hamsters were infected with a virus, and then they got an inhaler with these lipid nanoparticles. These vesicles containing the mRNA and the cRNA introduced into the lung cells of the hamster, where the nuclease looks for the virus RNA and kills it. And at least in the animal experiments, it worked very, very nicely. The virus load in the lung was strongly reduced, and I guess it's now in clinical trials, I'm not quite sure. It's highly promising. And not only for COVID, but for any viral disease that affects the lung, because you have to get it into the right cells, and the inhaler does not work on the liver or the heart, whatever. You can target the lung. We have to work on other delivery systems. That's one of the problems. Prime editing is another application. And here we are talking about the nickase, the nCas9, which cleaves only one strand. It's a little bit difficult system, and I will only briefly tell you about it. In this case, you attach an RT, a reverse transcriptase, to the nCas9, and you attach to the 3' end of the single guide RNA an additional sequence that represents the to-be edited part of the target RNA. So you have a very long sgRNA, which is extended. After cleavage of the target, now I guess I just show you the next slide where it's better explained, you have the Cas9, you have the target, you just cleave one side of the DNA, one strand of the DNA. This flips open, and we have sequences here that can anneal to the open end of the RNA. And here you have everything required for the reverse transcriptase. You can extend this 3' end of the DNA along the RNA, which is brought in by the Cas system. And you get double strand. And what happens then is you have when you re-anneal, when the Cas9 disappears, when you re-anneal, you have the choice between 3 strands. You have this strand, which can pair back here, and you have this additional one. So what happens? 2 different things. This one can be eliminated and cleaved off, the flap, and nothing happens. The edit, what you put in here, is removed. But it could also go the other way, that the original DNA strand is flapped out, and you establish base pairing with the edited site. And then the editing is enabled, it's included. We don't even have to go to this part. It looks like we have a 50-50 chance to get the edit, but we have now better methods to enhance this solution. It's not 50-50. This is much more frequent. Still not perfect, but it's much more frequent. The big advantage, you're changing nucleotides, but you're not cutting both strands. So you're much more targeted, you're much more specific. You do not rely on random repair, but you're doing something which is really targeted, and you provide sequences that should exactly repair the cutting site. So much better than the original. I guess I leave this out. I tell you about, because we are running out of time, right? I'll give you the link later. Okay. So there are new applications and improvements that are constantly being developed. I just go through a couple of applications. Plants and animals are coming on the market now, and there are lots and lots of them in the pipeline. You can introduce drought resistance, climate for dealing with climate change, CRISPR-based diagnostics. I showed you with the Cas13. This is an interesting thing that I haven't mentioned. In contrast to the conventional methods, you can do kind of multiplexing with CRISPR. So you can do a number of changes in just one experiment. The highest number, in as far as I remember, was 60 modifications in a single experiment. So targeting 60 different genes in a single experiment, and all get modified. In this case, you may have heard of David Bennett, who received a pig heart for transplant. Xenotransplantation is usually not working because it's rejected. But this pig heart was humanized in 6 genes, and it was not rejected by the patient. Bennett only survived for 2 months. I mean, he was terminally sick. He was going to die anyway, and he knew that. But surviving 2 months and not dying from rejection of the organ, but by other problems, is really a milestone in xenotransplantation methods. Well, just another application that's waiting, and it's a problem, I know, not only in Europe, but also in Indonesia, plastic pollution. Enzymes are available, but they are not very efficient. Modeling, improving the functionality of the enzymes will give us a great chance in the future to deal with plastic pollution. A major challenge is still the delivery, how to get the CRISPR tool to the right cells or to the right organs. Another challenge is how to make sure that there are no serious off-target effects. It's still, you always get or run the risk to get off-target effects. So I'm just pointing out the challenges for the future. In plants, animals, microbes, we have no problem to check if our operation worked as we wish. But when you do it as somatic gene therapy in humans, you can't do that. Yeah, you cannot throw away the humans where it didn't work and say, okay, let's take another one. You can do that with plants. You can do that with microbes. You can do it with animals, but with humans, no. So, in summary, we have achieved a gigantic progress within the last 10 years. And remember, CRISPR-Cas is 10 years old this year. Many things are already being applied. For others, we need further research and development. So synthetic biology is extremely important to drive these techniques, which will probably change the world and society. I can put this in the chat if you wish. There are many, or maybe somebody can put it in the chat, because I would really recommend these movies. There are several in the playlist. Check the playlist of the BioViscom YouTube channel. There are several movies that Khalid made for CRISPR-Cas modifications for CRISPR-Cas function, and they are nice. I really like them. And there's, of course, our BioViscom website, which is mostly in German, but it's easy to translate by DeepL. What else? We are just starting to also be present on Twitter, Facebook, Instagram, and LinkedIn. Join us there. Give us a like. Thanks for listening all this time, and I'm ready for taking your questions. Thank you very much.
All right. Thank you so much, Prof. Milan, for the very exciting presentation about CRISPR-Cas. And we already have some questions here. It's quite interesting that there are some people already give some questions.
Oh.
I will start first from Faisal from Biology Department of Islamic University Malang. So he has a question. May CRISPR-Cas9 possible to correct mutations at precise locations in the human genome in order to treat some genetic disease?
In principle, yes. You just have to get it there. So there are clinical trials with blood diseases where you can repair mutations, and they usually work. But these are clinical trials. It's not yet an approved routine technique, and it's extremely expensive up to now. But you can do it. You can, what is also a good target is the eye, because it's an isolated organ, and you can get with a syringe, can get close to the retinal cells. But liver may also be okay. But when it comes to heart or brain, then you have a problem, because you can't get specifically to the organ to cure mutations. So somatic therapy in some organs, in some cells, is okay. It works, and it will become routine, I'm pretty sure. In others, no. Very complicated to target the right cells, the right tissue.
Okay.
It will take time. Yeah.
Okay. And if I can continue the question,
sure.
because I'm a little bit interested also in your explanation. So what will be like the best delivery system then for in case of human genome or something like that? Do you have any insight of it?
It depends. I mean, you have to adapt the system to the specific problem. I mean, when you're trying to protect people from virus infection, you may introduce the Cas13 or the Cas9, depending on RNA viruses or DNA viruses. It depends from case to case.
Okay. So yeah, as you mentioned in your previous slides, you also show that the potential of CRISPR-Cas to be applied as like a vaccine using the lipid nanoparticles as well. And so there may be like one of example in how to deliver this system into the human body, maybe. Okay.
But as I said, with that you get you get the machinery into the lungs, but you can inhale very deep. You don't get it in your muscles, right?
Oh, yeah.
Yeah. So it's just keep in mind, this is not a vaccine. It's a therapeutic. It will kill the viruses that are there. It will not make you immune against viruses like a vaccine. Yeah, you don't trigger the immune system.
Yeah, so it's kind of a therapy.
Yeah, it's yeah.
Okay, thank you so much. So we can continue to the next question from Khalil. So his question is about the application of CRISPR-Cas9 in Indonesia. Maybe you can give your thought on this about maybe how far it will can be developed in Indonesia, maybe based on your experience as well. Yeah.
Yeah, it's a good question. The advantage of Indonesia, if it's still true what I learned a few years ago, Indonesian president has written a suggestion or whatever. It's an official paper where he asks scientists and everybody in society to work on genetic modifications when they are good for the people of Indonesia. So in contrast to Europe, and especially to Germany, where politics say, don't do this, don't touch it. Indonesia says, hey guys, go ahead, do it as long as it's good for the people. So that's an advantage. The disadvantage is people don't do it. I don't know why when CRISPR-Cas came up when I was last in Indonesia, it was already on. And I told people, hey, come on, it's not expensive. Get started. You don't have to do, to save the world, but just get it established in the lab. You have to get started, and then you can work on projects. And nobody really wanted to do it. I don't know what the situation is right now. If people are using it, it is essentially not that difficult, but you have to get it established, and you have to establish it in many places so people can exchange their experience. But that's one of the big problems in Indonesia. People don't really collaborate. But that's a different problem. It's not really so difficult to do when you're not aiming at highly sophisticated curing cancer. No, you will not be able in Indonesia to do that, because you're competing with huge labs in China, in the US, and what have you. But when it comes to improving your crops, for example, you can do it. You just have to do it. You don't get it done from just discussing. You get it done from going to the lab and doing the experiment. And this is what people should do in Indonesia. Yeah, it's not so difficult. It's cheap. It doesn't cost a lot.
Yeah.
Yeah.
Okay.
CRISPR, yeah. You know how much a CRISPR application is in, let's say, a plant or bacteria? How much does it cost?
How much?
Take a guess. A single application, a single experiment. How much does it cost?
In rupiah or in euro, maybe?
If you tell me in euro, it's better.
Let's say 50 euro or 100 euro?
Yeah.
All right.
50 to 100 euro for an experiment, and you have a modified organism.
Okay, so
this is so cheap. It's absolutely ridiculous. Yeah, just keep that in mind. Okay, you need more than one experiment, but even if you spend 1,000, it's not so much money. Yeah, but you have to do it. Okay, yeah. More questions.
Okay, thank you. So in terms of the diversity as well, I would like to highlight as well the biodiversity that we have in Indonesia. Yeah. Is that possible as well if we like doing a research, like trying to discover CRISPR-Cas system in our bacterial isolates, or maybe or something like that?
Yes, sure. I don't know how advanced whole genome sequencing or metagenome sequencing is in Indonesia these days, but it's not so. I mean, you can also do it yourself. And the sequencing machines are extremely powerful these days, and if you get a nanopore, it costs you 1,000. And I guess in the range of 500 euro per chip, and you can do a lot of sequences. Yes, sequencing and then collaborating with groups from other countries to do the analysis, the bioinformatics. I'm not sure, at least at the time when I was last there, in Indonesia, the bioinformatics was not highly developed. There was a lot of bioinformatics on proteins, but no really advanced bioinformatics on nucleic acids assembly or gene mining. That was not very well established. It's also easy. It's cheap. It doesn't cost a lot. I mean, for bioinformatics, you don't need a wet lab. You need a computer, and that's it. Yeah. And you need a couple of good friends in Germany or in the US or in Japan to help you run the programs and to establish the programs for gene mining. And that's it. Easy to do. That's what I told Indonesian universities 10 years ago. Establish bioinformatics, you'll publish papers. There's no way to not publish papers. With all the biodiversity, you have to find something which is unique. You will find something that is unique. That's a Nature paper.
Maybe, yeah, probably yes. Yeah, that's very encouraging.
No, I mean, let me just say this.
Yeah,
I told people, go to Kawaiigen and isolate or just take a couple of liters of water and sequence.
Yeah.
And you get a Nature paper. No, I don't want a Nature paper. I want to publish in the Bali Journal of Microbiology. Nobody's going to read it. Yes, but I get points from my university.
Yeah.
So now, I mean, you have to do it. You have to do it, and that's it.
Okay, so we still have quite a lot of questions here, like more questions pop out. So there is a raise hand from Mufliha, so I will let Mufliha turn on the microphone. Okay.
Terima kasih. Maaf saya pakai bahasa Indonesia ya Pak Hafiz.
Oke. Nanti saya bantu.
Iya, saya ini kan sedang akan melakukan penelitian tentang CRISPR berbasis biosensor berbasis CRISPR. Tapi yang saya pakai targetnya adalah DNA babi, dan yang saya gunakan jadinya bukan CRISPR-Cas 9 tapi CRISPR-Cas 12 karena dia menargetkan DNA-nya gitu ya. Kalau 9 kan bisa DNA bisa RNA dan dihubungkan juga dengan dia bisa ke biosensornya. Nah, yang saya tanyakan, dia kan akan memotong case-nya itu kan nanti diarahkan oleh track RNA-nya itu kan mengenali PEM-nya itu tadi ya. Nah, itu untuk memotong satu untai DNA-nya. Nah, untuk yang untai selanjutnya kan dia memotong di dua tempat nanti, bagian atasnya dan bagian bawah. Nah, itu dia spesifiknya bagaimana ya? Dia memotong di bagian mana yang di bawah itu? Kalau yang di atas kan arahnya tadi PEM-nya itu ya, ketemu PEM dipotong. Tapi yang di bawah itu bagaimana ya? Menempatkan dia potongannya itu di bagian sebelah mana untuk misalnya Cas 9, Cas 12 gitu.
Maksudnya mungkin di ini ya, apa namanya, yang negatif strand gitu ya?
Iya, karena
bawah universe itu ya.
Ya, yang dipotong kan dua jadinya kan. Dia langsung case-nya itu kan memotong dua, kalau di Cas 12 itu atas dan di bawah. Nah, lalu yang di atas mungkin dikenali dengan PEM tadi, si guntingnya akan memotong PEM-nya yang di atas, yang dia kenali PEM-nya di bagian untai atas. Tapi yang di bawah bagaimana? Si gunting tadi apa yang dia kenali? Dia menggunting bagian bawah itu di pengenal yang mana gitu di rantai. Ya.
Cukup itu, Bu Mufliha?
Ya.
Oke. So, ya, sudah bisa mungkin saya tanyakan ya. Oke. Okay, thank you, Bu Mufliha, for the question. It's very interesting. So, she's planning to do biosensor CRISPR-Cas based research of Milan, which want to use the CRISPR-Cas 12 system for targeting some DNA or RNA from swine. And then she has a question regarding to the target, the PAM sequence target of the Cas 12. So you already showed here, I guess, in your, especially this one, in your slide here, you mentioned that, this one, this is correct. So her question is, if we use the CRISPR-Cas 12, which is targeting like 2 strands of the DNA, And for the forward strand, let's say the CRISPR can easily identify the PAM sequence. But how about the negative strand, like in the bottom, like in the reverse complement strand? How to identify the PAM sequence in the negative strand DNA? Yeah.
So in this case, PAM is NGG on the opposite strand. So this strand is recognized by the CRRNA, and the opposite strand has the PAM sequence.
Okay.
Yeah, so, and this is recognized, and this implies when you have this sequence there, it's on the other strand, it's CCN, of course.
So it can.
But what?
Okay.
Yeah, it's a complementary strand, so it's a complementary sequence.
Okay. Okay. Yeah. So it doesn't matter if the PAM sequence is in forward.
No. The PAM sequence has to be on the opposite. The sequence which is the NGG 5' 3' NGG is on the strand which is not recognized by the CRRNA.
Okay.
So here is your target, and the PAM has to be on the other strand.
Okay. So the main thing is we should understand where is the PAM sequence.
Yes.
Right. Okay. Yeah. So it doesn't matter if it is in the forward or maybe in the reverse strand or negative strand.
It does matter.
Oh, okay.
Because you have to design your your CRRNA to the strand which is opposite the PAM.
Okay. So the CRNA should be in the opposite.
The CRNA goes to this strand and the PAM sits on that strand. Okay?
Okay. That's clear, I think. Apakah itu jelas, Bu Mufliha? Jadi intinya PAM sequence-nya lebih tidak di PAM sequence-nya. Kalau PAM sequence-nya di bawah misalnya, maka kita harus buat CRRNA-nya di opposite, di reverse complement ininya, targetnya. Apakah cukup menjawab, Bu Mufliha? Saya rasa cukup jelas ya. Oke, saya akan lanjutkan kembali.
Terima kasih, Pak Afif.
Okay. There are a couple of questions there still. I think I will just stop with the last 3 questions now. We have quite a lot of questions, I think. Yes. So I will start from Umu Syaukah. So she has 2 questions really. Okay. So the first question is, could you explain the difference between CRISPR-Cas9 and talents in case of specificity? So that's the first question. And then the second question is, do we still need a plasmid to help the transformation in CRISPR-Cas9? Yeah, you can start.
Okay. So specificity of talents, yes, talents will not disappear. They are highly specific, they are efficient, and there are a couple of clinical trials in humans where talents are still being used. The problem with talents is just that they are expensive, laborious, and take a long time to develop. And for every site, you have to construct For every sequence, you have to construct a new talon. Synthesis of an RNA takes overnight. The talons take longer. I mean, that's the major technical problem. I guess if you could make talons overnight, they would be as good or maybe even better than CRISPR-Cas. But the talons do not have all these other applications that I talked about. Yeah, so it's much more difficult to have all the other applications like diagnostic tools, like prime editing, like what have you, with talons. So they are not as versatile as CRISPR-Cas. Yeah, but they are functional and they will not disappear. It's just laborious to use them. And what was the other question?
Yes, do we still need a plasmid to help the transformation in CRISPR-Cas9?
It depends. When you want a transient activity of CRISPR-Cas, the lipid nanoparticles may be sufficient to target. But in some cases, for example, gene drive, I didn't talk about it, but it's an application which becomes extremely interesting. You want to insert things into the DNA, and I guess you need plasmids in most cases. We are moving in the direction that you don't need plasmids in some applications anymore. You can introduce DNA by other ways. But it will, in my opinion, I'm not 100% sure, in my opinion, I'd say plasmids will be around for some time before we get to a plasmid-free to plasmid-free applications.
Okay, the next question is from YouTube. So this webinar is also streamed live in YouTube, and there is a question there. So it's from Asrul Fanani. Yeah, his question is about how can I get the sgRNA prediction from the DNA? In his case, he would like to know if I want, how could I predict some sgRNA from the DNA of a plant?
Okay. Essentially, you can do it by just looking for PAM sequences and then designing your sgRNA from the site that you want to target. But there are also programs. I can't give you the link, but there are programs to predict sgRNAs. The linkers have been established and they are publicly available, so it's not magic. Search for programs for sgRNA prediction, and you find something.
Okay.
Sorry, I can't give you a program because I did not make notes on that. Yeah.
Okay, so maybe you can find in on Google. Start googling the sgRNA prediction program, especially maybe you can find for your specific case in the DNA of the plant. So the last question. Okay, it's from Advansu Herman. Cas13a, it's about Cas13a. So he asks that Cas13a cleaves single-stranded RNA, but mRNA may form secondary structure in the cells, and those prevent Cas13a to cut. What should I do to prevent this to happen?
Essentially, it happens. It just cuts. While it is absolutely sequence specific for the first targeted cut, it then is absolutely promiscuous. And all mRNAs have sites that can be cut. I mean, mRNAs are not indefinitely stable within a cell. They disappear anyway at some point. And this disappearance is strongly enhanced when you have a wild RNAs running around in the cell and cleaving everything. So secondary structure will not save a certain RNA. It will just be gone. And people who have been working with RNA know this as a problem. You isolate RNA, and when you look at it, it's gone because you have some nuclease or you contaminated your sample with some nuclease, and the RNA disappears within seconds, even though they have secondary structure. Yeah, it will just cleave everything.
Okay, I think that's a wrap. Thank you so much, Professor Wolfgang Nin, for your very interesting talk and also very exciting discussion session for today. And maybe do you have like a closing remarks from you?
Yeah.
So,
just thank you very much for inviting me. And what else could I say? It was fun. It was great to talk to you, and it was nice to see a couple of people in the audience that I remember from previous visits.
oh,
I hope to be back in Indonesia maybe next year.
all right.
And so all I can say is keep up the good work and make it even better. Get Indonesia into molecular biology. Good luck for Synbiotech to improve knowledge and applications of synthetic biology. I hope I could tell you a little bit about the chances, the developments, the applications of all these different CRISPR-Cas tools. And I hope they get established in Indonesia and getting used for agriculture, medical purposes, and whatever. So contribute, do it. Thank you.
All right. Thank you so much for your kind words, and also thank you so much for the participants who are joining us from everywhere. So I just noticed that we hit 155 participants in the Zoom today. It's quite.
That's quite good. Yeah.
Yeah. To end, before we end this webinar session, I think we have to take a picture, as usual, for the documentations. So please, for the participant, if you don't mind to turn on your video, so just turn on the video, and I will count for taking the pictures. So is anyone? So is everyone ready? Switch on the video. Let me check. Okay, but that's okay if someone's not available on the video. So we can just start. Okay, 1, 2, 3. First page. Okay, And the second page, 1, 2, 3. The third page, 1, 2, 3. And the last page, 1, 2, 3. Okay, so once again, thank you so much, Prof. Nilan, for your time and for sharing this very interesting topic. And we are looking forward to meet you in the next time. And also just a short announcement to the participants. There will be also a certificate if you want, but there's also some information that you have to know about how you can get the certificate. And yeah, so just stay tuned on our next program. Once again, for me, thank you so much for joining us today, and hope everything goes well for everyone. So thank you so much, and Assalamualaikum warahmatullahi wabarakatuh. Bye-bye. Have a nice weekend.